All right. We are starting the physiology of the male reproductive system. Two lectures are there. One is the male hormones, spermatogenesis regulation, and the second lecture is about the sexual act and the sexual performance of the male sexual act. Okay, so you will have that lecture also. One lecture is already there. I think we have to cover that lecture, which is about the mammary gland. Okay, we'll have to cover that lecture also, but that's not a major issue, inshallah. Look at the objectives. It's a shared lecture. It is between biochemistry and physiology. Now, the first objective is about physiology of male hormones, and second is about regulation. But inside, a hidden objective is that we must know about the biochemical nature and the characteristics of the male hormones. Okay? When we say male hormones, what do we include in these hormones? First of all, tell me. Okay, the top of the list hormone is testosterone. One. What else? We're talking about the male hormones exclusively right now. Okay, the male hormones only, exclusively. Only the male hormones. You said testosterone. What else? Yeah. Second is dihydrotestosterone, a derivative. What else? Androstenedione. Okay. Where is it coming from? Okay. What else? Prednisolone is an intermediary. It's a compound, an intermediate step. It's not a hormone. Yeah. Male hormone. One more. Yes, please. Yeah. Loudly. DHEA. Dehydroepiandrosterone. Now, if you classify them as testicular and adrenal, how would you do that? If I say, give me the top most important testicular androgen, what will you say? And if I say, go back to adrenal cortex, now you tell me DHEA and the second one is androstenedione. Okay, so let us take it like this. Testosterone is also coming from adrenal cortex, a little amount. Likewise, androstenedione and DHEA, they are also coming from testes, but as the head of the family, you will place testosterone here in the testes, and you will take DHEA and androstenedione for the adrenal cortex. They are all androgens, adrenal androgens or the testicular androgens. Dr. Mustafa will take over for the biochemistry portion, and later we will go to the physiology. All right?
So, as Dr. Wajid just mentioned, if we want to classify the androgens or the muscular male hormones, we classify it on the basis of their origin, the tissues from where they are originated. And we say that 90%, 95% of the androgens, they are originated from the testes, testicular androgens. And out of these, the most abundant is testosterone. A variant of testosterone, which is called dihydrotestosterone, is also there, and it is most potent, more potent than testosterone. But the abundant one is the testosterone, and also from testes small quantity of androstenedione. But majority of the androgens that come from adrenal gland, they constitute about 5 hormones. Out of these, the most abundant is DHEA. Now come to the testicular androgens. The cells that synthesize these androgens, they include Leydig cells. Clear? And they are synthesized, their synthesis starts at the level of mitochondria in the cytochromes. Clear? The immediate precursor of this hormone is cholesterol. They are steroid hormones. So being steroid hormones, they all are synthesized from cholesterol. So the synthesis starts in the Leydig cells at the level of mitochondria in the cytochromes from cholesterol. Clear? And the main influencing factor that initiates this synthesis is luteinizing hormone from anterior pituitary. Clear? So in the first stage, the cholesterol converts into pregnenolone. This is the first step. This happens under the influence of LH, luteinizing hormone. Then the pregnenolone undergoes two set of pathways. One is called delta 4 pathway and the other is called delta 5 pathway. Delta represents the presence of double bond. If the double bond is present at position number 5, this is called delta 5 pathway. If the double bond is present at position number 4, this is called delta 4 pathway. So if you see, in the first stage the cholesterol converts into pregnenolone, then pregnenolone undergoes a series of reactions. Clear? And converts into androstenediol. All these steps, they are categorized as delta 5 pathway. This delta 5 is just the location of the double bond. Clear? On the other hand, the pregnenolone can be converted into progesterone in the initial phase, and then this progesterone undergoes a series of chemical reactions, and this is called delta 4 pathway. And in the end, if you see, you will get testosterone. Clear? So to remember this pathway, you don't need to remember the steps, but what you need to remember that the main factor that starts the synthesis is cholesterol. The first reaction converts the cholesterol into pregnenolone, which undergoes two sets of pathways. One is called delta 5 pathway, the other is called delta 4 pathway, and the end product is testosterone. Clear? The cycle starts in the mitochondria. Clear? This is one thing. In testes, in human testes, the majority of the pathway that is followed for the synthesis is delta 5 pathway. Clear? Sir, do you want to add anything? Okay. Sir, feel free to add anything wherever you like. Okay. As I told you, delta 5 is the most important pathway, more important. And after the synthesis, the testosterone, it converts into dihydrotestosterone. Dihydro indicates, the name indicates dihydro, addition of two hydrogen bonds. So when you add two hydrogen bonds in testosterone, the testosterone converts into dihydrotestosterone. I told you the most abundant hormone that is synthesized is testosterone. It is converted into dihydrotestosterone because dihydrotestosterone is more potent, more active. Clear? And because this reaction requires addition of hydrogen, so the enzyme that catalyzes this chemical reaction is reductase. So now we discuss these two hormones, testosterone and dihydrotestosterone. As I told you, majority of the hormone that is synthesized is testosterone. Its quantity is 5 milligrams per day, and dihydrotestosterone, its quantity is 50 to 100 microgram per day. This is microgram. Clear? So if you see, it is 100 times less. So once they are released from the testes, they enter in the circulation. Because of their lipid nature, steroid nature, they cannot circulate freely in the plasma. Clear? Only 3% of the hormone is circulated freely. Rest of the hormone, it binds to the proteins, plasma proteins, and majority of the protein to which they bind to sex hormone binding globulin. Clear? And also they bind loosely to albumin. Clear? So in blood you find them bound both to albumin, and the binding is loosely, and to a specific globulin which is called sex hormone binding globulin. Which form is the biologically active form? The free form or the bound form? The free form is the active form. Now, after they are released in the blood, they circulate in the blood for about 30 minutes. Clear? Now, after they bind to the target tissues for their actions, Dr. Wajid will describe their target tissues in detail and their actions. Clear? So we are skipping the part of the actions and we are moving forward for metabolism. Clear? So they bind to the receptors in the target tissues. After their binding and execution of their actions, they are metabolized into inactive forms so that they can be excreted out of the body. Clear? One metabolism which is very important in this category, that they are acted upon by the enzyme aromatase. Clear? And they are converted into their ketone derivatives, and these ketone derivatives, they are excreted out. Similarly, in liver, the oxidation also occurs at carbon number 17, and again they convert into their ketone derivatives, and these ketone derivatives, they conjugate as sulfates or they are conjugated as glucuronate, and they are excreted in urine. Clear? Is it clear?
After they fix the target tissue.
This is their action. This slide, this will be explained by Dr. Wajid. When they bind to their target tissues, what happens? Sir, can you just go through this slide?
Okay, so because you have asked the question, you will see that from circulation, the free hormone reaches the tissues. When you are saying tissues, tissues is a collection of cells. It means you are going outside. So testosterone is reaching outside the cells. Okay. Now testosterone will enter now into the cells of the tissue. Okay. Now it is intracellular in position. Okay. Inside the cell, we believe that there is a special enzyme called 5-alpha reductase. Okay, 5-alpha reductase. So if this 5-alpha reductase is present, that will convert the testosterone into its reduced form, and that form, Dr. Mustafa already mentioned, is dihydrotestosterone. Okay. Well, some of the testosterone remains unconverted, as testosterone. The other part is converted into dihydrotestosterone. So now inside the cell, you have two hormone forms. One form is the original testosterone, and now a modified, more active, more potent dihydrotestosterone. Now they will act according to the needs. Both will have an action because these are steroid hormones. Do you remember the action of the steroid hormones? They are acting through the nucleus. Both will have their receptors, but the final action, the potency of the action will be different. Now in some of the tissues, this 5-alpha reductase is present in large amounts or in large quantities. Okay? That tissue will show a dominance of which hormone? Dihydro or testosterone? If you have alpha reductase molecule in large amount. Okay. And some tissues have a little quantity of alpha reductase, so their testosterone remains the major form. Okay. So I'm not saying that 100% testosterone is modular in one cell or 100% dihydro. Actually, it's the proportion which is present here. So as a dominance, if dihydrotestosterone is there, you will see there are some tissues where you will have a better action of the dihydrotestosterone. Can you identify the tissues from the slide? What are the main tissues where you have, muscle. For the dihydrotestosterone, its action. Prostate is one. Okay. What is second? External genitalia is number 2. What else? Skin. Remember these three ahdaf, the most important target area, the skin of the individual. The second thing is the external genitalia, their formation and their development. Okay? Right from the fetal life, that you are seeing human being, you are considering human being, human life from the fetal life. So when the fetus is developing the external genitalia, which is more important, dihydrotestosterone or testosterone? Oh yes. When the fetus is developing prostate gland as an internal genitalia part, which one is more important, di or definitely? when the fetal skin is developing here, or distribution of the receptors of the follicles, so who is controlling more, diet or? So the skin changes. The internal genitalia, khususan prostate, and almost all of the external genitalia, their development and growth and function is controlled by dihydrotestosterone preference. So that's why you say, okay, the enzyme alpha reductase, which is 5-alpha reductase, that is found in large quantities in these three areas especially. In other parts of the body, other cells, it is not true. Little amount. Okay? The testosterone is active. Okay?
Okay, so after their action is finished, they need to be metabolized. Clear? And this portion of the slide, it is explaining their metabolism, where the testosterone is converted into an inactive form so that it can be excreted out. Clear? And this metabolism mostly it happens in the liver, where the oxidation occurs at carbon number 17 and the testosterone converts into androsterone and dehydroepiandrosterone, and then these two hormones, they are conjugated either as sulfates or gluconates, and they are then excreted out in urine and stool. Clear? Clear? Now the testosterone, if you see, majority of the testosterone are for, not majority, like 20% of that testosterone, it acts on the Sertoli cells for spermiogenesis. And in the next part of the lecture, we will discuss this topic in detail. Clear? 80% of the testosterone, it acts on other tissues, as we just mentioned. And you should remember the 3 or 4 tissues like skin, external genitalia, prostate, muscles, liver, where they act. Clear? And one more thing in metabolism of testosterone, that testosterone can convert into estradiol. I hope you have seen this conversion during the synthesis of estrogen. Clear? So one very important aspect of testosterone, that it converts into estradiol. And what is the enzyme that catalyzes? Aromatase. Clear? So this is very important conversion. You should remember this. The testosterone under the influence of aromatase converts into estradiol. And as I just mentioned, the concentration of testosterone is about 10 times more than the dihydrotestosterone, but dihydrotestosterone is more potent. Now comes the regulation, sir.
All right. Okay. So if you've got a picture of the hormones, their distribution, and you have also seen what happens when the hormones have done their action, their job, and they get metabolized in different tissues. Okay. So it's about the metabolism also. Having done the metabolism, few points about the regulation. The principles of regulation are just the same. It's an axis which you already know, mashallah. There is nothing new in it. You have hypothalamus in the axis on top of the list, and then you have in the middle the pituitary, and finally it's the gonad. Here, because we are discussing the male system, we are talking about the testes. Look at the axis, gonadotropin releasing hormone coming down, and it's controlling 2 important hormones, the same hormones for the females, the same hormones for the males. Males, they have got their own characteristic actions. In females, FSH and LH have their separate roles. Okay. Now you are considering the role of FSH and LH, the same female hormones. That's why it is wrong to call FSH or LH as a female hormone. Call them as gonadotropins. Gonadotropins is a general word which can be used for the male or can be used for the female, isn't it? Okay. So be safe. When you are saying what is FSH or LH, what will you mention? They are just gonadotropins regardless of sex. They are active in both sexes. They are equally important for us and for the females. Let us see what are they doing in us. The FSH and LH, you know that FSH is coming down and it is acting on the testis. Now, where in the testis?
LH is also coming down and acting on the testis, and you have to find out the specific location. So let's take FSH first. What is your idea? When FSH is coming to the testis, what will be the hadaf? What will be the target of that? Testis is a big area. So what do you think? FSH, where will it go? What will be its prime action, the prime cell or the prime area? What do you think? Sertoli cells. Thank you very much. You know, in the testis, if you go back to the histology, are you done with the histology of the testis? Yes. There are two very, very special prominent cell types. One is Sertoli cells, the big cells or the giant cells, okay? The tough cells, long living cells, resistant cells, very robust and very, very stable cells, okay? They are mechanically supportive, they are kind of connective tissue cells, but very strong cells. These cells are unique in their characteristics. We'll talk about that. But right now we are talking about FSH is coming, and FSH is acting on the Sertoli cells. Now what is FSH doing for the Sertoli cells? Okay, FSH will find its receptor on the surface, no problem. Okay, what will happen next? Inside, it will induce the metabolism of testosterone. Dr. Mushtaba just mentioned that 20% of the metabolism of testosterone is there. It is meant for the Sertoli cells. It was there in the previous slide if you saw. It means that testosterone is converted into a living active pathway, and that is known as the aromatization, isn't it? You are further aromatizing the testosterone by the use of which enzyme? Aromatase. All right. So are you acting on the testosterone? If you are going back to the previous slide, what is happening here? Testosterone is getting converted into, where is this action happening? Testis. Where in the testis? Sertoli cells. You are inside the Sertoli cells now. Imagine yourself inside the Sertoli cells. So it means, what is FSH doing, by the way? It is, forming the estrogen, estradiol from. So what is the principal action of the FSH again? It is inducing a very special enzyme. It is increasing the quantity of an enzyme. Which one? So its focus is to act on the aromatase and stimulate it. When you have more aromatase, you will have more conversion of testosterone into estradiol. This is aromatization. Now, aromatization of the testosterone here, is it feminization or masculinization? What do you think? Generally speaking, you are forming a female hormone from a male hormone, isn't it? Okay, so in general the process is known as feminization. Okay, but if you see the philosophy of this process, this estrogen formation is not making us feminine, it is actually making us masculine. Okay, this is the beauty, this is different. Okay, if in other parts of the body you are converting testosterone into estrogen and estrogen and estrogen, what will happen? We are changing into a female like structure, isn't it? Our body is changing. But here in the Sertoli cells, something very special is happening. Okay, the process is apparently feminization. The enzyme is feminizing enzyme. You are producing a female hormone, but that female hormone is fascinating and very, very important for us, because without this female hormone we cannot form our sperms, final forms of the sperms. So for the proper formation of the sperms, you need this estradiol. Okay, when you are finally changing the sperm and you are having the morphogenesis and forming the tail of the sperm, giving it shape, giving it complete morphology, you need this little estrogen. This little estrogen is so important for us. It means our fertility depends on this little amount of estrogen. Look at the importance of this action, which is done by the FSH, aromatase, conversion, and finally on the sperms. Okay, we'll see further. So that's why I reminded you that this process is very important. Okay, this is required for all the males, for normal functioning of the males, without which the fertility will not be there. Okay, all right, this is one thing. FSH is doing its job. Okay, all right. In response to FSH, definitely you have to give the message back to the pituitary also, isn't it? So the Sertoli cells are also doing one more thing in response to FSH. When they know FSH has arrived and FSH is working, then the Sertoli cells will also produce one more special feedback factor. And that factor is a little hormone which is coming from Sertoli cells. And that is an inhibitory hormone that will tell the pituitary that enough FSH is mojood, now we don't need more FSH. So is it negative feedback or a positive feedback? So what do you think? It's an inhibitory hormone. What should be its name? Inhibin. In the males, in all of us, inhibin is coming from Sertoli cells. Okay? So FSH is doing a double job. It is actually, first of all, triggering the process of estradiol formation, and it is also informing the Sertoli cell that I am here. So you also produce inhibin, and that inhibin will leave and it will go back. Okay? So the feedback inhibition. This is the feedback loop for the FSH. GnRH to FSH, FSH to Sertoli cells inhibin, and inhibin back to the pituitary. It is going to the gonadotropes. Is it okay? Now let us take the part of the LH. What is LH doing? Okay? Now LH, you remember its role in the females. Khalas, keep it aside. Now consider the males. Well, are you with us? Are we all together? I don't think so, please. Okay? LH. GnRH to LH. No problem. LH is now released. There is no cycle. There is nothing like female cycle that in the middle of cycle you will have LH. Not in males, okay? We don't have any cyclic pattern. We have the release in fluctuations, but it is not cycle that 14th day or 18th day or 20th day you will have LH release. La. Okay, males are different. Okay, males are males. So LH is coming. LH is also coming to the testes. Dr. Mujtaba mentioned one more cell type which is very important, and these cell types are very sensitive. Sensitive in the sense that these cells are quite vulnerable in nature. They are easily destroyed by effects and insults from surroundings like radiation, drugs, toxins, chemicals, antibodies, and many things can destroy these little cells. Very important cells, very interesting cells. They live in clusters. They like to live in the groups. They like to live in interstitium, so they are interstitial cells. Because they were discovered by Leydig, they are called the interstitial cells of Leydig, or the Leydig cells. Leydig cells are lying outside the seminiferous tubules. If you have the seminiferous tubules like this, okay, between the seminiferous tubules are the islands or the areas where you have the clusters of these cells. Okay, so Leydig cells are very special cells. They are hormone producing cells. Your principal androgen, which is, oh yes, so your testosterone, in testosterone, males are producing testicular androgens from Leydig cells. LH is acting, receptors are present on the Leydig cells. Okay, protein hormone, receptors on the surface, stimulation and production of the testosterone. Testosterone will be produced mitochondrially, as Dr. Mujtaba was telling us. Final formation by the pathways, 2 pathways, Dr. Mujtaba told us about that. Okay, and testosterone is released. Now, after coming out, testosterone has to decide really that what should I do now? Okay, I have to take care of the local area, dakhli area, which is testes, because testosterone has to control many things. Some of the testosterone will enter into the Sertoli cells. What will happen there? It will be converted into, this is one of the jobs of the testosterone. Some of the testosterone also has to act locally, but it will say that no, I have to go into the seminiferous tubules because sperms need me. I have to support the sperms. Okay, so again via Sertoli cells and outside, the testosterone will find its receptors in the growing sperms and in the Sertoli cells, so that it also controls the process of spermatogenesis. So seminiferous tubules, dakhli action. Sertoli cells, dakhli action. The major quantity, remaining quantity of testosterone will diffuse into the blood, and it will go systemically into the bloodstream, and it will circulate. Now it has to perform functions in all parts of the body. So it means testosterone has to take care of the testis, functions of the testis, and the functions elsewhere in the body. All right? So this way, this LH, which was controlling testosterone, now testosterone is in the bloodstream. Testosterone will perform all the actions in different parts of the body. Muscles, metabolism, skin, hair growth, whatever, all the actions, okay?